Welcome to a Chapter 14 virtual lecture. We're going to finish up this chapter talking about how a cell regulates cell division. Earlier in this chapter, we discussed mutations. You've also read about cancer and its progression, but how do we reach that point? What we're going to see in today's virtual lecture, there are a series of changes that must occur in a certain subset of genes. If those genes are targeted by mutations, they can ultimately lead to the loss of regulation in the process of cell division. So let's test you out. Let's see how you've done in preparing the chapter. So we discussed in Chapter 9 the idea of cell signaling pathways. In a cell signaling pathway, we often have an extracellular signal that binds to a receptor. If you recall, that receptor then is activated. It transmits the signal throughout the cell, often using a signal transduction cascade that included kinases, and ultimately that's going to cause a change in cell physiology. We saw a particular pathway which was mediated by the Ras protein, in which we saw two kinases, MAPK and MEK. So now in this question, if you have mutations in the normal MAPK and MEK genes that resulted in abnormally high levels of kinase activity, resulting in an increase in the rate of cell division, the mutated gene that's result is called a what? So the genes for MAPK and the genes for MEK have been altered. Now their kinase activity is actually accentuated by that mutation. What type of gene is that considered? That's an oncogene. We're going to see oncogenes result from gain of function mutations, in which we see an increase in protein activity, or we may see an increase in protein levels, all of which are going to accentuate the desired result, in this case, cell division. We're also going to look at a second class of genes called tumor suppressor genes. Tumor suppressor genes stop, the products of these genes stop cell division. In many cancers, we lose the activity of tumor suppressor genes, so we would consider them loss of function mutations. All right, so let's define oncogenes. Now we know that cell division is a highly regulated process. It has to be. When you lose control over cell division, what often results is cancer. We know that cell division can be regulated by hormones called growth factors. These are typically extracellular in nature. So what they do is they bind the cell surface, causing a change in that receptor, leading to that cascade of events, which includes upregulating gene expression. We've seen this before. This is all review. Now, if we have mutations in genes whose products signal cell growth, we call those genes oncogenes. Oncogenes promote cancer by not allowing cells to go into G0. We talked about G0 previously. It's the resting state. Most cells are in G0. They're in the pit stop. However, if these oncogenes are mutated and upregulated, what results is a cell that's continually signaled to go through G1, S, G2, mitosis, cytokinesis. Now, how can an oncogene be turned into an oncogene? Why is it on? Now, it could be that the gene product is produced at high levels. So, for example, this can happen in receptors. If that receptor is made at an artificially high level, ultimately it's going to be sensitive to even small amounts of growth factor. What that means is the growth factor doesn't have to wait in line. It would really coat the cell, and all of those receptors that are bound are all signaling to the cell, hey, let's divide, let's divide. So that's not so much a gain of function mutation as it is a gain in gene product mutation. And in other cases, we can actually tweak the enzyme activity to work better than it did before. So the kinase activity can't be shut off. And as a result now, if you can't shut off the gene product, now again we're constantly signaling for the cell to divide. Here's a typical signal transduction pathway. This is the RAS pathway we talked about in Chapter 9. Please go review if you don't remember. Here was a tyrosine kinase receptor. So as a result of a growth factor being in the environment, it can bind to the receptor. The receptor changes shape, causing it to autophosphorylate. That autophosphorylated receptor then activates these bridging proteins, which ultimately causes RAS to get rid of its GDP and exchange that with GTP. When RAS, the monomeric RAS protein, is activated, it turns on kinases like MAPK and MEK, and those particular proteins can then activate transcription factors, which ultimately increase the products that signal cell division. So, what represents oncogenes? Now know this, folks. In their normal state, they're called proto-oncogenes. I mean, I need my cell to divide, but I only need it to divide when signaled to. So the gene for this tyrosine kinase, the gene for RAS, the gene for MAPK, the gene for MEK, heck, even the genes for these transcription factors, all of those are proto-oncogenes that are only activated in the normal signaling pathway. However, if those genes undergo mutations, which would cause a rise in gene products, or if we have a mutation that would increase the activity of the protein, that would then cause a proto-oncogene to transform into an oncogene. An oncogene is the gene that's responsible for overstimulating cell division. So what can be an oncogene? Any of the genes here. The gene for the receptor can become an oncogene. The gene for RAS can become an oncogene. The gene for MAPK and MEK, the genes for the transcription factors, all of those have the potential to become oncogenes if the mutation leads to one of these two scenarios. So let's look at RAS. Now, RAS has been implicated in many of the well-known and well-characterized cancers. When you look at the mutations that need to occur for cancer to progress, RAS is one of those common oncogenes that's involved in a wide and diverse range of cancers. Now remember how RAS worked? RAS worked by when it signaled, we lost the GDP and exchanged it for GTP. And now RAS can go signal to the rest of the cell. But just as importantly, it needed to have phosphatase activity. RAS needed to hydrolyze the GTP to turn itself back off. If it can't turn itself off, we can imagine that active RAS is going to continue to tell the cell to divide. So an oncogenic mutation in this case would result from a loss of function mutation in which we've lost the ability to hydrolyze GTP. So here's where the mutation would occur. And as a result, the signaling pathway is always on. And if the signaling pathway is always on, what we end up with is unregulated cell division. This is really one of the first genes. There are many, many more that have been characterized. I think there's about 400 genes that are involved in the progression of cancer. But RAS is one of the first to be well characterized and well documented. It's been well studied. So now, why does this happen? How can a normal proto-oncogene transform into an oncogene? Well, the four most common genetic changes are missense mutations. Now, we talked about that. A missense mutation results from a change in the DNA, which results from a substitution of an amino acid. And that change at a particular position could result in either increased or decreased protein activity. So we've looked at mutations from the level of a gene coding sequence, but now we can start to take bigger pictures. There are other common changes like gene amplification. That results in an increase in the gene dosage. So remember, we talked about an increase in the number of protein products could lead to uncontrolled cell division. Well, think about this. If a gene is amplified, if there are many copies of a gene, now we get just that much more protein. And with that much more protein, we can have a cell that's very sensitive to growth factors, which causes unregulated cell division. We can also have chromosomal translocations. I'm going to show you a great example of a rare form of leukemia. I call it now the Kareem Abdul-Jabbar leukemia, since he's been recently diagnosed with this, in which fragile chromosomes can actually exchange genetic information. So what we end up with is a totally new chromosome. So part of chromosome 2 can break off and fuse with part of chromosome 8, and as a result, we have new gene and we have new regulatory regions. And then we also know that viruses can integrate themselves. We know that certain viruses can take their RNA, transcribe it back to DNA, and that DNA actually integrates to a host cell chromosome. Well, we'll show you an example of how that can lead to an oncogene. So missense mutations are fairly straightforward. We talked about these. Now, could you define what a mutagen is for me? That's right, a mutagen is an agent that increases the rate of mutation. Now, they could be chemical agents, cigarette smoke, for example, alcohol, for example. It could be physical agents like UV rays. We know, for example, that chemical mutagens found in cigarette smoke can lead to changes in sequences that affect the activity and, in some cases, the expression of proto-oncogenes. And now, what may happen, for example, in the case of RAS, is that missense mutation results in the loss of GTP-ACE activity, which results in a pathway constantly being on. Now let's talk about gene amplifications. Often what can happen is we can have slippage where we replicate a gene a few times. Rather than a single replication, we have multiple replications in which polymerase can slip back. What results in this case is an abnormal number of genes. Gene dosage goes way up. Let me give you an example. Now the MYC gene, the product of MYC, is that MYC is a transcriptional activator that increases the rate of expression of proteins involved in cell division. So now you can imagine if MYC becomes duplicated and we have 3 gene copies instead of 1, we're going to theoretically now have 3 times the amount of MYC protein in the nucleus. And the more protein you have, the more gene expression you ultimately end up with. And the more gene expression ultimately leads to increases in cell division. We have seen this kind of gene duplication in a variety of cancers: breast, stomach, lung, colon, brain, even human leukemias. All of these have shown MYC genes to be amplified. Other genes that would lead to oncogenic expression, other genes when mutated by gene amplification, would be those genes for receptors. This is a very well-known case in certain breast cancers in which there is so much protein receptor produced that what you end up with is a cell that's constantly stimulated. That can come about via gene amplification. So I don't think that concept is that difficult to sort of wrap your head around. Right? The more gene copies you have, the more expression you ultimately end up with. Now let's talk about chromosomal translocations. In this case, when chromosomes break, we can have fusion of those 2 broken chromosomes, and as a result, we end up with these chimeric genes that may have, for example, a change in the regulatory region. So for example, here's a normal proto-oncogene. Here we have the coding sequence, here we have the regulatory region. Now could you imagine, for example, that if two chromosomes broke off, part of which then brought the coding sequence, the other chromosome brought its own regulatory region. And what would happen now is if this regulatory region is much more efficient. What if it's expressed more often? Because it has a better KB or KI. Well, what's going to result now is this proto-oncogene is going to be expressed at much higher levels. And as a result of that overexpression, we lead to an increase in the rate of cell division. For example, we have the translocation, these two genes, the BCR gene and the ABL gene, they confuse, forming what we call a chimeric oncogene. This is a rare form of leukemia that results from this. Here's the example. Here we have our normal chromosomes. Here we have breakage at the bottom, so now we have an exchange where the BCR is exchanged here, the ABL is exchanged here, and what we end up with is what we call the Philadelphia chromosome. This is not part of the normal 23, it's a whole new chromosome. And as a result, we get increased expression in this myelogenous leukemia because of a change in the regulatory region causing a proto-oncogene to be overexpressed. Last one here, retroviral insertions. A retrovirus has the ability to integrate its own DNA into a host cell chromosome. Now again, here's a normal proto-oncogene with its normal regulatory region. You can imagine in some cases, if we get insertion within that regulatory region, bringing in new, more efficient sequences from the virus, what can happen is we can enhance expression, ultimately then resulting in an increase in products that transform it into an oncogene. So we can see mutations are a very general term. Mutations can arise in a variety of ways. A change in the coding sequence at the missense level, a change in the number of genes, a change in the chromosome structure, and even bringing a sequence outside the organism. So we can see we keep broadening our scope as we get from one level to the next. In each case, what results is an overactive oncogene, which increases the rate of cell division.